Welcome MS translators to another one of our MS Virtual 2020 videos. And it is my absolute pleasure to be joined today by Dr. Will Brown from the University of Cambridge. Now it's really exciting to have Will here with us because Will actually gave the presentation at MS Virtual 2020 around the Bexarotene trial that we did feature already briefly in terms of sort of coverage. So this was a remyelination trial, and Will's been very polite, I guess, in agreeing to talk to us today about it and give up his time. So thank you, Will. Can I start maybe by asking you to just give a bit of an introduction to yourself and your overall research interests?
Well, hi, and thank you for having me. I'm a senior neurology trainee in Cambridge. I've done my clinical training between London, the National Hospital for Neurology at Queen Square, and Cambridge. As you know, I came out and spent some time at University of Melbourne doing a real-world evidence fellowship with Thomas Kalinchik.
So I guess the main reason that we reached out was to talk about your presentation at MS Virtual 2020. So can you tell us a little bit about the trial that you discussed there? Just a general overview of it to start with.
So we did a phase 2 trial in 50 people with relapsing remitting MS, which was using a drug called Bexarotene. And this is a drug that's currently licensed for a type of blood cancer called cutaneous T-cell lymphoma. And this drug acts on a particular receptor called the RXR gamma receptor. And so we wanted to find out whether using that drug in comparison to placebo promotes remyelination.
Okay, fantastic. So obviously it's an area of huge interest in the MS research community at the moment, and certainly people living with MS are really interested in it as being the next big step in the treatment of multiple sclerosis. So I'm sure there'll be a lot of interest around this. So can you tell us a little bit about what was the background to this trial? So what led you to choosing Bexarotene as a candidate for this?
So the trial rests on the work from Robin Franklin's group, so he's a professor in Cambridge. If anyone wants to look at the sort of hard science paper, it was published in 2010 from Jeffrey Huang in Nature Neuroscience. And to cut a long story short, they looked at the different stages of repair, and let's just go right back to basics. So putting the myelin back onto nerves after it's come off because of inflammation. We know that myelin can go back on its own. That process is called remyelination, but it seems to fail, and particularly with age. And so Robin's group showed that there are a number of potential mechanisms behind this, but the one with the greatest effect seems to be by the blockage of RXR gamma. And so this receptor just seems to stop working in animal models. And they found in a rat model that when you then agonize that receptor, when you stimulate it, the myelin goes back on. And they did the same thing in human tissue as well, in some cerebellar slices. So the background is they've used a drug to prove that remyelination can work by agonizing this receptor. And Bexarotene is the only licensed drug in humans that also agonizes that receptor, that stimulates that receptor. The downside to it, which we knew before going into the trial, and we'll come on to, I'm sure, later, is that this drug is not specific. So this drug agonizes all types of the RXR receptor. And we know that when you stimulate, from Robin's work, the gamma subunit, then that's what puts the myelin back on. But when you stimulate the alpha and beta subunits, then that's what gives you the nasty side effects, and we certainly saw those.
Okay, fantastic. So I guess then the natural lead-in question is, so you've gone through and you've tested this compound in this trial. What were the outcomes that you saw?
So the most important outcome is that this drug is too dirty to use, and that was really disappointing. So everyone who was randomized to Bexarotene developed an adverse event, and in fact, on average, they developed 6, which is very high in any trial. Every patient developed hypothyroidism, so reduced production of thyroxine, and all of them who stayed on the drug ended up on thyroxine replacement. The vast majority, I think 92%, had elevated blood fat levels, so blood triglyceride levels. And then there were a number of other side effects, including half of people developing headaches, half of people developing a rash, and a third of people dropping their neutrophil count, a type of white cell you need to fight infections. So this is not a safe drug to be using. There was one particular case, or that perhaps the most worrying adverse event that was seen, was that one patient was found to have a silent stroke on their MRI scan at 6 months. And just to be clear, that means that we do the scan, they haven't had any symptoms of a stroke, but there's definite evidence of a stroke there, and we don't know whether that was due to Bexarotene or not.
Okay.
So that's the kind of the safety side of it.
Yep.
What's more positive is that the drug has evidence that's most likely remyelination. So to go from the top, these studies, there's a very limited number of remyelination trials at the moment. But of course, these have all been running for years. They've all been designed years ago. And when they were set up, we didn't know how to measure remyelination. We have a better idea of it now, but as a result, we picked an MRI scan outcome as a surrogate, and with hindsight, we probably wouldn't have picked now, but of course we stick to our protocols and report what we say we were going to report. So our primary outcome was actually negative, and we did not find that the change in a particular scan sequence called MTR that correlates with myelin, that there was any significant difference between the lesional MTR in placebo compared to the lesional MTR in Bexarotene. And that seems to be for 2 reasons, and this is kind of a crux of what we found. First is that we'd expected to find a difference, as it happens, of 1.2 percentage units. And that's what we were powered to detect. But actually, what we saw was a difference of 0.16. So we were never going to see a statistically significant effect. But the second thing is that this makes an assumption, which is that all lesions on an MRI scan have the same capacity for repair. And there's growing evidence showing that that isn't the case. And so the next bit of our analysis was to look at the literature before we actually started analysing this and say which lesions are likely to show an effect best. And based on that, we actually did see some highly significant effects, some in particular groups of lesions, which is biologically plausible. So, sorry, rather long answer, but in short, we show that on MRI, our primary outcome is negative. But we have shown that in particular groups of lesions that we know from the biological literature are most likely to show an effect, that there certainly is an effect. And then the last section was we use visual evoked potentials, which is a way of measuring how quickly a nerve impulse passes from the back of the eye to the back of the brain. And that directly correlates with how much myelin's on those nerves. And we showed that there's a statistically significant improvement in the Bexarotene arm, whereas the placebo group continued to worsen. So I think, in short, we've shown that there is an effect.
Yep. Okay. And so, I mean, I think that it's obviously really interesting results, and there's so many ways that you can look at the results of a trial like this. And so, obviously, the main outcome here is that the trial hasn't worked. I mean, that's sort of the model in terms of the safety, and it hasn't met some of those primary outcomes. But I want to talk to you a little bit about the future directions and the framing of this, because I think it's really important. And when I originally posted one of the articles about this trial that came out during the conference, I didn't go into details with it, but I think that framing is really important because the headlines around it, which you might find surprising based on it being essentially a failed trial, was that the MS treatment is a step closer, was one of the headlines that we read a lot. And while I think that's going to be surprising to people when they hear, the results were in essence negative, there is a lot that we can unpack from that. So can you talk to us a little bit about, one, why that headline still makes sense, but sort of the future directions now, what this leads us into?
Yeah, I think this is critical. So the first legacy that we hope comes from this trial is saying that this mechanism of action does seem to work. So that by stimulating this particular receptor, this RXR gamma receptor, with a first trial that shows converging evidence from using visual evoked potentials and MRI to support, to provide evidence of remyelination. That hasn't been done before. And this is important because it now puts a lot of emphasis on finding a drug that will just agonize that RXR gamma receptor without the alpha and the beta subunit agonism. And drug companies are trying to do this. So that's the kind of that to me is the most important thing here. And with any luck, that will come to fruition, and the benefits that we've seen will then come without the adverse events. But I think the second side of it is also how to do a trial. This is not an easy thing, and I say that more from the point of view of the participants. We don't want to subject people with MS to these kind of side effects. I do not think we would have done this trial if we knew the adverse events were going to be like this. They were far more frequent than they were in the lymphoma literature, and it was a very uncomfortable trial to run, and we're incredibly grateful for the participants who stuck at it. But in terms of how we make these trials better, we now know which lesions to look at with MRI. And I'm quite sure that this will evolve, and we are continuing to do other analyses, as are other groups. And we're also finding which tools to use non-MRI, so for example, with visual evoked potentials. So I think that the two legacies from the trial are, one, that RXR gamma is a good way to promote remyelination. I think we've got really robust evidence of that, despite this being a negative trial. But secondly is how to run a trial, and all lesions on an MRI are not the same.
Yeah, and that's it. I mean, it's such useful information to be able to use, as you say, moving forward. It's a jigsaw, and I think we keep getting these puzzle pieces and working out how to fit them together. And this is another one of those that's going to lead to hopefully eventually positive trials, hopefully in the not too distant future. So there is another trial that's going to start in the not too distant future, I guess. Can you tell us a little bit about that one?
Yeah, so people will know about the clemastine trial from Ari Green's work that was published in The Lancet in I think 2019. Is that right?
I could be right now. I can't remember the year.
I think it was.
We'll post in the comments under the video if it was wrong.
Yeah, please correct me. And this study was the first to show a statistically significant effect that is likely remyelinating. They found nothing with MRI, but they did show a significant effect with visual evoked potentials. So the second trial from the Cambridge Centre for Myelin Repair is going to be a phase 2 trial again, and it's going to compare 25 people who are randomized to receive clemastine. But in addition, they're going to be given metformin. and then compared to 25 people on placebo. And the rationale for this again comes from Robin Franklin's group, who've shown that there's a paper that came out a year or two ago looking at alternate day fasting in animals. And this showed that this can promote remyelination. But we know that translating that into humans wouldn't be feasible. It would be unsafe to ask people with MS, and we really do not recommend fasting. Because it will lead to weight loss, it will lead to muscle loss, and that will have a negative outcome. So please, we strongly recommend against this. However, in the same study, Robin's group looked at the drug Metformin, which is a drug used in type 2 diabetes, and it had the same effect, or it had a comparable effect. So, and it looks as if Metformin is particularly good for allowing. So to take one step back, we know that in MS, the problem is not a lack of stem cells to create, to then differentiate or change into cells that can put the myelin back on, so into oligodendrocytes. We know that it's a problem with turning them from the baby cells, from the OPCs, into myelinating oligodendrocytes. And so Bexarotene, clemastine, Metformin, they seem to target that aspect of this, but Metformin seems particularly to do this to aged cells. So the theory is that by giving Metformin and clemastine together, it's going to have a sort of a super added effect. It'll be a synergistic effect.
Okay, fantastic. Yeah, so I mean, I did a video summary of the fasting Metformin paper that we put across MStranslate, and I think it's one of the most watched YouTube videos that we've ever put out. There is a huge interest in that area, so I'm sure people will be interested in finding out more about that, the Metformin part of the trial, once that becomes available. So probably a question that's a lot of people maybe already know the answer to, but I think it's still important to discuss. With these remyelination trials, probably in general, the potential outcomes of these for people living with MS. Can you talk about that a little bit?
So I think you've alluded to the progress that's been made with anti-inflammatory therapies, and particularly for people with relapsing remitting MS, but growing evidence in progressive disease as well. And there is now this big shift towards how to repair. And that's right, because I'm quite sure that there is more work to be done with anti-inflammatory effects in terms of, in particular, working out which to use when and in whom. But most effort is now being channeled into looking at how to repair. And so I think these remyelinating therapies are the most exciting way forward on this, for numerous reasons, but at least not that it protects the underlying nerve. So it doesn't only allow the nerve to function better, but it also protects the nerve. And that means that when nerves are lost, when nerves die, that is the biggest kind of correlation with disability. So to answer your question, what do these remyelinating therapies hold for people with MS? Well, in the long run, the hope is that these will reverse disability. And no studies so far have been big enough to examine that in any meaningful way. So phase 3 studies of successful drugs from remyelinating therapies at phase 2 would be looking to do that.
Okay, yeah, fantastic. I often refer to this as, at the moment, our stage is almost like you've come home and you've found that the pipes in the basement have sprung a leak and your basement's flooded. And at the moment, the stage that we're at is that we're managing to plug those holes in the pipe so that we can stop more water from flooding in, but we're still leaving all of that water in. And really what we're hoping to do with these remyelination therapies is start to remove that problem area that lies there in the damage. So a super interesting area. I guess lastly, and we've probably covered a little bit of this as well, and you've already agree completely with you in terms of thanking all participants on trials, because it is a huge sacrifice that they make to be involved in this, and there's people in the research community. It is much appreciated. But is there anything else that people with MS and the MS community can be doing to support your work and the work of the Cambridge group in general at the moment?
I think, as you say, sort of keeping interested and keeping asking us questions that that make us think, and for those who are nearby, keeping participating in our trials. It's a huge ask. We really recognise this, and it's just a huge admiration that we have for people who do come forward and offer to be involved in trials. I think it's a positive experience overall. That's the feedback we get. It's a nice chance for us to get to know people as well. But people often find that they get to sort of ask us lots of questions along the way and sort of find out a bit more about stuff they haven't had a chance to with their normal healthcare providers, just because research settings have a bit more time. I guess if anyone is so this trial, the second trial with Clemastine and Metformin, will be in recruiting only in Cambridge, and I know that limits the number of people who will be able to participate. But keep an eye out on Twitter. So the handle is Cambridge Neuro 1, the number 1, and we'll certainly let you know when we open recruitment. That should be just after Christmas, COVID dependent.
Okay. All right. Well, fantastic. It goes without saying that I think the admiration that you have is also returned. I think the MS community in general will be super appreciative of all of the hard work and dedication that you and the rest of the team have towards this research, and also for giving up your time to share this research with the MS community. It is much appreciated by all of us. It's much appreciated by me. So thank you for doing that, especially knowing how busy you are to spare the time to do this. We're really thankful for that. So thank you, Will, for this. And we look forward to talking to you again soon in the future.
Lovely. Anytime. Thanks, Brett.
Thanks, Will.